So today I'm going to talk about my research, and first of all, thank you for this opportunity to present at the GOTBIN seminar. And my main focus of my today's presentation is computational method to explore host-parasite interaction. Next slide. Few words about my background. I am from the Ural Mathematical School, and I was fortunate to learn from great scientists like Nikolai Krasovsky, who made a great contribution to game theory, and Professor Lev Shevrin, who is an outstanding algebraist. And during my bachelor studies, I focused on computer science and math, and we had supercomputers, so we can try to apply mathematics to solving different applied problems like in medicine, biology, economics, and social sciences. I took one course in bioinformatics as an undergrad, and for me it was a turning point. I was fascinated by how similar concept and algorithmic appear in biology. And since then, I became more and more interested in bioinformatics and how we apply computational method to different research questions in biology and medicine. And then I go back to academia for my doctoral degree. My advisor is Professor Daniel Bowley, and he shared and supported my interest in recognition system and biology. And we developed methods that allow us to apply set theory, linear algebra, and metrics method, and also artificial intelligence method to different research questions in biology. And today I focus on a couple of our key questions. Next slide. The roadmap for my talk, I'll talk a little bit about overview of viruses. And usually when we talk about microbiome research, the role of viruses is often overlooked. Because usually when you hear the word microbiome, you're thinking about bacteria, commensal bacteria or pathogenic one. But viruses is like hidden but very important component of this microbiome. And I hope to convince you to focus on the role of viruses in microbiome research by showing you 3 examples that for me change my perception of viruses. And in my third part, I talk about the theory and application of computational method applied to virus-host interactions. Next slide. Next one. Microbiome is very diverse. It includes not only bacteria, but also archaea and fungi and protists. And all of these organisms have their own viruses. So the diversity of viruses is really great. We have human viruses, animal viruses, plant viruses, fungal viruses, also special viruses that can infect bacteria and archaea. And all of them are present in our intestine and in our body. Next slide. Next slide. The first example that I want to talk about is how bacterial viruses can trick human immunity. Next slide. This is an example of one of the human pathogen, Pseudomonas aeruginosa, which is a gram-negative bacterium that also is present in chronic wounds. And many of these clinical isolates are antibiotic resistant, and there is no vaccine available for this infection. And many of the, next slide, many of these strains has an integrated prophage in them. And it was interesting that strains with and without the prophage demonstrated significant difference in terms of outcome for patient. Next slide, please. So here, the analysis of human ones infected with Pseudomonas aeruginosa with and without this prophage. And you can see on A and B, significant difference in terms of the appearance of the ones and time for healing in the graph in section D. So this makes a huge difference. Next slide. If you're interested, I encourage you to look closely into this research, but the main take-home messages that I want you to get from this paper is that this prophage can be released by bacteria and it can trigger human immunity. If you look at the left, this is what normal immune response in the absence of prophage. But if the prophage is present, then bacteria can release this prophage. And on the section on the right, you can see that this prophage activate antiviral response in human cells and steal resources that can be otherwise distributed to fighting the bacterial infection. Next slide. Interesting that this provides the opportunity to develop vaccine against this, to reduce the severity of infection. So this vaccine is not against human virus, but it's against the bacterial virus. And it can significantly affect the time to healing and the amount of infection present. And I think, next slide, please, that similar approaches can be taken with respect to other infections. And I think vaccination against prophages might be preventive for those as well. It has a broad utility in chronic 1 infection, cystic fibrosis, and also immunocompromised cancer patient. Next slide, please. As well, it might work for Salmonella and Escherichia coli as well. And also these findings can complicate phage therapy, because now you can, I hope you can see that this viruses of bacteria is a double-edged sword, because they can help bacteria to trick human immunity, and also they can help human in killing the bacteria. Next slide. The second example that I want to show you is how viruses can restore healthy gut microbiome. Next slide. This is the original article. It's this research about murine virus. Next slide. So in this research, it was shown that murine virus, it's eukaryote virus, unlike the previous example that I show you, but in germ-free mice, researcher observed some defects in the formation of the healthy intestine. And when they introduce this single virus, that can significantly reduce this deficiency. So it kind of it could replace this healthy microbiome to some extent and provide necessary immunological stimuli for the intestine to develop properly. Next slide, please. And this showed that it doesn't have any effect in normal mice with a healthy microbiome, because they don't have this problem. However, next slide, please. Next slide. So in this case, viruses can play a positive role as the compensation for deficiency in healthy microbiome, and I think it's very important for antibiotic antibiotic treatment when healthy microbiome can be depleted, so we can use viruses to compensate for those negative changes. Next slide, please. And the last example about the positive, interesting and significant role of viruses is viruses in cancer microbiome. Next slide, please. So previous research demonstrated that there is human-specific viruses that are strongly associated with cancer. However, the role of bacterial viruses was not determined. So this particular research uncovered some aspects of how phages can contribute to the virome during for oncological patients and how it can contribute to cancer. Next slide. So this is a really huge problem in US because about more than 1.5 million Americans are diagnosed with colorectal cancer, and over 500,000 died from this disease. Next slide. And the question is if there is an accurate and non-invasive detection, preferably from the stool samples based on the bacterial and viral community composition. It will allow us to have early detection and prevention of colorectal cancer. And also, what can we do to use the bacterial viruses to help eliminate this unhealthy microbiome that also can provide a burden for immunocompromised patient. Next slide. So here, this study had 90 human subjects in 3 different stages of cancer. And 16 RNA genes, shotgun sequences was analyzed, and viral particle was analyzed, only those ones that was included within viral capsids. Next slide. The researcher tried to find the best model performance that take into account the composition of microbiome, including viral and bacterial composition, and see which one have the most predictive power. And it turned out that the combination of bacterial and virus composition had the best performance in this case. So definitely virus have some information that can be missed if you analyzed only bacterial impact. Next slide, please. I don't want you to capture the entire picture right away, and if you're interested, I refer you to this paper. But here the main idea is that bacterial viruses can alter the microbiome, and it can be beneficial either for cancer progression, or if we understand this impact, we can alter it in a good way. So I hope that by that time I convinced you that the role of viruses is really important. And that's, next slide, please. Also, it was interesting that this research showed that the major role in this disease progression played what they called influential phages, or phages that can affect multiple species of bacteria. So by targeting these influential phages, we can alter the community and reduce the burden from cancerous patient. Next slide, please. So these 3 examples that we just discussed show you that viruses do play a significant role in health, human health and animal health and disease. That's why I become really interested in exploring this virus-host interaction, particularly those ones that exhibit themselves in exchange of genetic material. Next slide. First, I'll tell you a little bit about the theory. And here I want to, if you look at the middle picture, this is how I imagine the genome, how it looked like in all that complexity. But when we try to analyze only part of the genome and try to figure out what genes are, it's resemble to me this picture on the left, when we neatly stack all component that seems similar to us. But really, if you have only this picture to the left, it's hard to go back and recover all the complexity that we observe in the complete genome. That is why in my research, I focused not on a particular gene, but on analyzing complete genomes, so genome as a whole, without eliminating any fragments of the genome, just based on the fact that we don't fully understand what they are. And not a gene, it doesn't mean it's garbage. It means that we don't fully recognize the significance of this particular fragment. Next slide, please. So because we're talking about the at least two types of organisms, bacteria and viruses, and how they interact through genomic exchange, we can formalize this in the following, in terms of set theory. So you can imagine this all possible set of strings that we can observe, and some of these strings appear in bacterial genome, denoted here in yellow, and some of them can appear in viral genome that appear in blue. And this intersection in green demonstrate the fragments that are shared between bacteria and viruses. And these fragments can be an important sign of biological interactions. The question is how we can capture these signs reliably, because on one hand, there is biological significant interaction, but on the other hand, there is shared fragments that are just due to the fact that all genomes have this restricted four-letter alphabet. And some of the fragments can be shared just due to random coincidence. And for me, it was interesting to understand how this intersection changes if you vary the string length that you analyze. And next slide, please. So if you, here is just some mathematical notation to simplify the exposition. So here G is a genome, and cardinality of G represents genome size, N is a string length, and U sub M is a set of all possible strings of length M. And the cardinality of this set is 4 to the mth power. It's what we call the complete dictionary. And the size of this dictionary grows exponentially if you increase the string length. D sub M of G is a set of string of length M that is present in a particular genome that we will call genome dictionary. The question is, what is the relationship between this size of genome and size of these two dictionary as M varies? And can we utilize this knowledge to distinguish between spurious and biologically relevant genomic interaction? Next slide. Here I give you an example of a bacteria, Escherichia coli. It's a lab strain of bacteria. It's not pathogenic, and a particular virus, FX172. And the graph on the right show you how the size of the genome dictionary for these two organisms varies as we increase the string length. The gray diagonal line represent the size of the universal dictionary or the complete dictionary of strings. And you can see that because this, as M grows, we have a lot of diversity, and different organisms can occupy different subsets of this universal set. And only biologically relevant organisms continue to share long fragments if we increase the string size. Another important observation here is the first fragment, which is straight line, where all the blue, red, and black line correspond to each other, is when we usually see the K-mer analysis, because, or substring counting. Because for this method, what is important is the frequency of strings. And to analyze the difference in frequency, you need to have all the strings present in a genome. But at the same time, this area is very noisy, because a lot of, there is not many unique strings present in a genome, and the set of the strings shared between two genomes is usually the same. And to reduce this noise, the K-mer techniques usually try to come up with different methods to discriminate this noise. But my point is, if you increase the string length, then you can eliminate this noise naturally, because the probability that two unrelated genomes shared very long string is very small, given the exponentially growing size of the complete dictionary for this given length. Next slide, please. So here is a brief summary of previous findings in terms of strings analysis and their frequencies. It's a very useful method, but the shortcomings, as I mentioned, is that we have spurious relationship that need to be discriminated. It's interesting that long strings is successfully used for taxonomy identification, for example, in Kraken, M equals 31. So this, the use of this unique string allows us to classify reads from metagenome samples. However, we still don't know. Most of these findings are still empirical, and we don't know why certain lengths work better than others for a particular data set. And another problem is that when you have findings for a given data set, you cannot generalize it. And if you have switched to a different data set, you need to repeat this string search analysis again. Another question is, what are major factors that affect the choice of string length in biological system? Because there's biological systems, for example, CRISPR-Cas system, that use exact matching, and usually spacers have a length in a particular range, from 25 to 40 base pairs. And the question is, why biological system choose this particular range of lengths? What is so special about them? Next slide, please. So to analyze this, to make this dictionary computation, the existing methods were not suitable because the majority of them were optimized to make this substring counting efficient for large metagenome samples. And in this respect, it was really hard to add some additional parameters or change the data structure, even though the algorithm complexity of constructing this dictionary is linear. So in this case, you can use simple hash tables implemented in Python for the exploratory purposes. And that's exactly what we did. And to avoid the bottleneck of uploading large genomic data for analysis, we start to test our method using the synthetic genomes. So it has several benefits because you can vary the content of a genome, and also you can vary the length, and you can discard them when you no longer need it. But when we compare the result of the analysis for dictionary-based method for synthetic and real genome, we found a very interesting regularities. Next slide, please. So here I show you synthetic and real genome. And on the left, on x-axis, it's Streamlines M, and on y-axis, it's a semi-logarithmic scale that represent number of unique strings in intersection. So here we take a real pair. It's E. coli, it's a pathogenic strain E. coli, and it's phage type 1 on the left and type 10 on the right. And then we take this pair and simulated this genome using random number generator. And we generate about 10,000 runs for each pair. Other curves showed you mean intersection in 10,000 runs and maximal intersection observed in 10,000 runs. And in panel A, you see that the intersection observed between synthetic genome is indistinguishable from the intersection observed for the real pair. But if you look on the panel B, there is a significant difference, because for the randomly generated unrelated genome, the intersection disappears for lengths more than 25 in different 25 in different runs. But at the same time, the intersection between real genome persists for much longer lengths that cannot be explained by random coincidence. And this long tail is what can be a reliable sign of some biological processes. And so p-value is very significant because it's 1 out of 10,000. And if we want to increase confidence, we can run this simulation multiple times, for example, million or 10 million times. Next slide, please. So the question is how we can capture, it seems that the previous slide showed you that there is might be a threshold for spurious overlaps in terms of string lengths, but how we can capture this threshold when we change the genome, the size of the genome or composition of GC content. Next slide, please. To better understand that, we apply statistical modeling. So we screen different combination of lengths for viral and bacterial genome, and also different level of skewness in terms of GC content. And here I show you 3 bands of the results. The first, the panel A, demonstrate case when the genomes are skewed in opposite direction. The middle band represent case when both genome, viral and bacteria, has the same uniform distribution of nucleotides. And C panel demonstrates the case when both genomes are skewed in the same direction. And you can see that this curves from probability of observing spurious overlap. It moves as you vary GC content and length. And there is a regularity to it. So longer genome and similar skew shift the threshold to the right. For us, it was important because in this case, you can try to come up with a theoretical estimate for the threshold, because it looks like there is some regularities to it. Next slide, please. And we develop a theoretical formula that allows us to compute the probability of spurious overlaps based on the probability of a single match. So here I'll briefly walk you through this, but if you're interested, I encourage you. At the end, I have a link to my doctoral dissertation where I go through this computation in more details. So here you can imagine two genomes as a generator or die that have 4 sides, and when you roll it, it gives you either A, C, G, or T with the probabilities that correspond to the given genome. And because we are talking about 2 genomes, we want to understand what is the probability to get the same, get a match on those dice. And any match, either A or C or G or T, will surface. So that's why we multiply the probability of observing A in one genome by the probability of observing A in the second genome, and we sum up because any match will do. This gives us a probability of a single match. But what is probability of observing no match between 2 strings of length m? Is 1 minus p to the m. And if we add another level of complexity, so now you have 2 sets of strings. One has n1 string and another has n2 strings. And we're interested to understand what is the probability of observing no match between these 2 sets of string. And this is exactly what gives it by this capital P at the bottom. Next slide, please. We computed this probability of no match using the probability of observing different nucleotides in the genome. And these probabilities can be estimated using the information about GC content. And if you plug this GC content for genome 1 and 2 in this formula, you got this equation that match the probability of a single match to the GC content in both genome. And if you plot this surface, you can observe a saddle point that corresponds to the probability of 1/4 when both genome has the uniform distribution of nucleotides. At the same time, we have local maximum of 0.5 for the probability, but this is not reachable in the living system because it corresponds to the case when both genomes have extreme skew of either 0 or 1. It means that both genomes can have only C's and G's or A's and T's and doesn't have the other nucleotides. But the question is, what is the most extreme GC content reachable in a genome? Because we can use this estimate to compute the threshold. Next slide, please. So to understand what is the reachable level of skew, I analyze all the sequence genome available at the NCBI, and the lowest GC content in prokaryotes and viruses is 13.5%, and it is found in insect symbiont that has very rather small genome, about 200 base pairs. And when we plug this skew case, we can come up with a maximum probability for a single match that is reachable in a real genome. And if we plug this probability in the formula for observing no match, we can estimate this probability for different string lengths. And that's what is shown on the right, in the table on the right. So for this case, you can see that the probability of no match rises from 0 for very short strings, because there is always something in intersection because this of spurious match, to almost 1 for very long strings. So for strings of length 40, there is nothing that you can see due to spurious overlap between two genomes that correspond to the usual range of lengths for prokaryote and viral genome. Of course, if you want to transfer this knowledge to the eukaryote genome, like human or plant, you need to make adjustment because the size of the genome will be significantly different. But it's possible to make this estimate using the formula that I just showed you. Okay, next slide, please. Okay, so here I show you how we use this threshold for evaluating the biologically important and spurious overlaps. And we can use it to make different predictions. Next slide, please. So here I show you the results of screening between a set of phages that contains about 2,000 different phages and 2 E. coli. One is lab strain on the left, and another is a highly pathogenic strain on the right. And to be able to compare intersection between phages that have different size, we introduce the relative measure. So it's amount of strings shared between two genomes divided by the size of the smaller dictionary. In this case, this gives you a value between 0 and 1. And that's 1 correspond to the case when the entire viral genome is inserted in the bacterial genome. Like, for example, this gigatoxin producing phage that is completely present in E. coli 100 or 157H7. And observing this graph, you can see a striking difference between interactions with phages between lab strain and pathogenic strain. The question is it possible to predict functional properties of host based on this intersection with phages? Next slide, please. And here we test this hypothesis using the set of phages and 101 different E. coli strains. This data set is balanced because 57% of these strains were pathogens and 43 were other, like commensal, laboratory, and biotechnological strains. Next slide, please. For screening for intersection, we use this length 40 computed theoretically, but the question is how we can evaluate sensitivity and specificity of this screening. So to do that, we use we compute the number of phages with non-empty intersection for different screening window. And you can see that if you choose very small window size, like below 30, then you observe intersection between E. coli with almost every phage, even the one that doesn't infect E. coli. So there is a lot of noise. But if you increase the window size, you observe this plateau. And if you increase the size even more, then you start to lose the signal. So this theoretical computed threshold of 40 seems to be reasonable in capturing enough, capturing signals of intersections with enough sensitivity and specificity. We also use the same screening window to compute intersection between these two E. coli and a set of eukaryote viruses. and we found only 2 cases with non-empty intersection. And you can treat this as a false positives, which is very low level. But at the same time, when we dig into these 2 cases, it turned out that it's not a false positive because there is a biological reason for observing these shared fragments between bacterial strains and human viruses. Because, for example, this vaccinia virus was developed using the E. coli as a vector. So part of the E. coli genome was present in this virus just by design. Next slide, please. So we found remnants of no less than 30 phages within each E. coli strain, and we used this intersection with this set of phages as the feature vector. And based on this feature vector, we tried to infer the pathogenicity of a particular bacterial strain. We use random forest for binary classification, and out-of-bag error was about 14%, which was good. And the question was, is it possible to reduce this set of phages to a small subset of indicator phages that you can use to screen newly sequenced strains of E. coli and get an estimate of probability for it being pathogenic or not? Next slide, please. And the answer to this question is yes. So using feature selection and ranking, we found a set of 6 indicator phages that can be used for quick typing of bacteria or distinguishing them on pathogenic versus non-pathogenic. And this is a rather complex slide, but I try to explain you. So we have on the bottom, we have E. coli strains arranged based on the probability of being pathogenic obtained from the model. And also it's represent the true labels. 0 means it's non-pathogenic and 1 means it's pathogenic. And 6 stripes on the top represent the contribution of different phages in this prediction. And you can see that these phages usually contribute to pathogenicity, but they capture different aspects of pathogenicity, and taken together, they can capture it better. So each of them contributes its own type of information for predicting pathogenicity. And accuracy of this subset of indicator phages was about 91%. We use 5-fold cross-validation, and this gives you an average estimate and standard deviation. Next slide. So if you're interested to learn more about dictionary-based method, here is my doctoral dissertation published. And also we present this part, including hands-on experience on machine learning on microbiome data. And here is some materials, including data set and some scripts that you use as hands-on experience to try this yourself. Okay, next slide. Thank you for your attention, and I would be glad to answer any questions.